I'd like to introduce a true friend of the LHON community. Dr. Alfredo Sadun is a world-renowned specialist in neuro-ophthalmology and is at the Doheny Eye Center, UCLA. Dr. Sadun has developed an international reputation, especially for diseases of the optic nerve, such as Leber's hereditary optic neuropathy. In 2012, Dr. Sadun was recognized with the highest clinical distinction in neuro-ophthalmology, the Hoyt Award, presented at the American Academy of Ophthalmology in conjunction with the North American Neuro-Ophthalmology Society. Dr. Sadun. Rustum?
Well, Rustum or you figures out the computer. I'll speak a bit about what a pleasure it is to be here. There are a lot of meetings I go to, but I can't think of any other meeting that has as much energy and enthusiasm as this one. And so I was trying to figure why that's the case. I mean, these are the same scientists I sometimes see at other meetings, and I think it's because of you, because they're being reminded why it's so important what they do all the time, and so they take it that much more seriously. So specifically, I wanted to thank three groups. One is the UMDF for having allowed this to happen. Are we there? And it's really a great meeting, as I was mentioning. The second is to you, both as you sit here in this room, but many of you are my patients. And as Rustum sort of alluded to, it's a big deal to do the clinical trials. It's a heavy burden on both doctors and patients. Well, it's our job. But the fact that you as patients have seized some control of the situation and saying you're going to be part of the solution is really a wonderful thing. And we almost abuse our patients. I see them there for 10 hours in one day, one test after the other. And not only do you guys put up with it, you do it with a smile. And that means, again, a lot for us. But thirdly, very specifically, I have to thank Lissa. Lissa has been nothing short of magnificent. The energy she brings, the intelligence she brings, and I guess most of all, the dedication she brings to this whole process is very much, in my mind, in my little corner of the world about Leber's, very much part of the solution. So with that, I thought I would do a quick review, a few minutes of Leber's, and then mostly about what's on the horizon. And I'm very glad that Rustum had a chance to explain to you about clinical trials, because we're going to have a test at the end of this session. No, well, we will refer to his work regularly with some of the terminology, and you've made my life a lot easier in that regard. So, with that, I should start off with the financial conflicts. One of the things that's important for us when we're looking at this is to say, what biases might we be under? So, for example, I'm going to be talking to you about a half dozen clinical trials, but 2 of them I'm doing myself. Working with GenSight and Stealth Biotherapeutics, we're running clinical trials. So it's going to be a natural, and hopefully I'll fight the tendency to lean in their direction and say, this is really the good stuff. So it's important to throw out there, oops, you're not seeing that very slide, to throw out there the fact that GenSight and Stealth Biotherapeutics are in fact funding these clinical trials I'm speaking about, that I work with at Doheny. I lost it all, huh? That's true. You know what? Listen, I think that we're going to go ahead. So, one of the reasons that there's been so much progress recently in Leber's, and I'm not being cynical, it's just the reality, is because it is a dramatic disease. And being a dramatic disease, many of the drugs that can be tested in the setting of Leber's can be assessed more easily. Back to one of the points that Rustum made. I would not be surprised if we will make the first discoveries in Leber of a very powerful agent that helps mitochondria that will then be used for diseases that are much, much more common, maybe Parkinson's. And so it's understandable that a drug company that would say, gee, it's going to cost me $5 or $10 million to do a clinical trial that's going to help this tiny community, will also be saying, but now I've got an agent that I can bring to market big time. So we have, in fact, achieved a great deal of attention from these. We're working fine now. Thank you very much. From these groups. Wasn't me. I haven't even touched anything yet. So the nature of Leber's, as I said, is dramatic, but highly specific, not necessarily to Leber's itself, but to mitochondrial diseases of the optic nerve, something we've been calling MON. And the features I like to emphasize, this is a slide I often recycle, is that it's bilaterally symmetric. Whatever happens in one eye ultimately happens to the other with tremendous predictability. You start off usually with a loss of color vision. The field defects are central, and this is because of anatomy. The fibers involved are the ones that deal with central vision, and they come off the temporal side of the optic nerve, so that in all these diseases, not just Leber's, but the other genetic diseases, the other nutritional diseases, the other toxic diseases that all seem to injure the same pathways, they produce this particular feature of the side of the disc involved. So because most often when I go around, people vaguely remember having read a book 20 years ago that had the word Leber's in it, and that's all they do, I try to give them a flavor to the disease by describing the typical patient that I see in the U.S. And this is often a college student, and maybe he's doing a little bit more smoking and drinking than he has before because he's off at college. But while at college, loses vision in one eye very dramatically. Goes to the infirmary where they say, I don't see anything in the eye. But they send him to an ophthalmologist who does, who says, aha, young person, sudden loss of vision, and there's certain tests that can be done by the ophthalmologist that locate it in the nerve, even though it's not visible. And he says, 99% of patients who have an optic nerve problem who are about 20 years old, it's optic neuritis. And I won't even take a vote, but I would imagine that the vast majority of people in this room, before they had the diagnosis of Leber's, had the diagnosis of optic neuritis. And the vast majority of patients who have optic neuritis have MS. And so usually the patient is told, good news, bad news. Bad news is you have MS. We've got to do a bunch of tests to prove it. Good news is there's not only therapies for MS, but most of the vision comes back. But then the tests are negative, and then the other eye goes, and that's unexpected. So eventually they find their way to a neuro-ophthalmologist, of which there's only about 150 or 200 in the country in academic settings. So it's not an easy thing to find one. But the neuro-ophthalmologist usually instantly gets it because they've seen that pattern before. Now, for fun, I always like to say Brazilian presentation is different because it's usually not a college student, and a few other things are different. And maybe if anybody's really interested, we can discuss later on what makes it different. This slide, for those who can't see, it has a little subtitle on the very top called the slide of lies, or sometimes they call it the slide of little lies, because it's not exactly true, but it simplifies the situation. And it says that you've got maybe 20 or 30,000 genes in your chromosomes. That's the major inheritance that you get from your parents. But you've also got these tiny little bean-like structures in every cell in the body that have their own genes, 13 of which make their own proteins for the special metabolism that the mitochondria have to do. And the mitochondria seem to play two significant roles. One is to make the energy for the cell, the ATP, and the other is to make the byproducts of that process called reactive oxygen species. Now you might think, because you hear all the time about antioxidants, that the reactive oxygen species are terrible things, and they often are. But they're also good things. They're messaging systems that are being used within the cell to say how they should alter the metabolism. But they're the generators of a lot of their own. So that's the endogenous ROS. Now if you smoke or do other things, you get some more ROS from the outside, and sometimes that tops off the tank. This next slide is actually the pathways, so it doesn't have the little eyes in it, and it's just a little bit more complex. But one of the things that this slide illustrates is that there's an electron transfer chain. And electrons from complex 1 and 2 get shifted by this quinolone to 3. And then 3 goes to 4. The point being that there's two ways to get an electron to 3. And that means that you can have a defect of complex 1 and still the system can operate. And that's what Leber's is, a defect of complex 1. So in one sense, Leber's is the mildest possible damage that can occur to the electron transfer chain because you can't work around it. And indeed, there are other genetic diseases that are more severe that usually produce more devastating consequences. But if you inherit a mixture of mitochondria, half good and half bad, you either don't get those other diseases because the good mitochondria carry the load, or you get a much milder version of those diseases and it looks just like Leber's. So I have patients of mine whose genetic defect is actually MELAS or is actually Lee's disease, but to me it looks like Leber's because they only have a mixture and a diluted effect. So in one very strange sense, you're lucky. If you have Leber's, you got the mildest thing that can happen to the electron transfer chain. I can't help but revisit Brazil when I talk about the subject, because that was an opportunity of doing two things. Most importantly, of seeing over 350 patients in this gigantic pedigree that taught me a great deal about it. And two is to bring together people who were friends of mine to start with, but now have become family, because living for a week in remote rural Brazil every year, we get close, and they're in the front row right here. And so we have worked very much shoulder to shoulder in both these trips and in general on the process of dealing with labors. And the lessons of Brazil I sort of distilled down to 2 or 3 slides. Let me teach some terminology first, although you probably know it well. If you have the gene, you're just a carrier, just. If you have lost vision, significantly you're affected. So the first thing is, and genetically those two groups are identical, of course. The first thing is that affected people may lose vision rapidly, as we talked about typically, but it doesn't stop. Unaltered, without proper support, it keeps getting worse. The textbooks miss that fact, because after 2 years of an ophthalmologist telling his patients, there's nothing I can do for you, the patients stop coming back. And so the ophthalmologists don't realize that by the 5th year, they're worse. By the 10th year, even more so. So we have followed these patients for 15 years, and we can see that. It's also hard to see because it doesn't affect visual acuity. And visual acuity is the cornerstone of what the ophthalmologist uses to measure. So you go down 20/20, 20/30, then 2400, and then we usually go to counting fingers, and then we stop measuring. And the patient may keep getting worse, but he's still counting fingers. And the way he gets worse is about the visual field. It's a big central area that the edges just keep moving further and further out. And if you can measure that, which is hard, you can see the progression. The second general take-home message from Brazil involves those who are asymptomatic, who have no visual complaints, but they're not perfect. The carriers often have subtle abnormalities. 1/4 of them, you can see them directly by looking in the back of the eye. There's this area where the fibers are compensatorily swollen. That is to say, they probably are stacking up with extra mitochondria because they know there's a little bit of a problem, and that compensation works fine. And if you do a very sophisticated set of color tests, not the ones that ophthalmologists usually have, half of these patients can be shown to have a little defect. And if you use OCT, they're probably the single most useful tool that we have in our toolbox. Then you can see, especially in the men, that the same thing I mentioned, that there's a little bundle of fibers especially that pick up extra mitochondria and therefore swell. And this is actually an illustration of what, in the old days, including us, when we first saw this patient, we said, that's normal. And then by the 2nd or 3rd year, a few of us were saying, not quite. And then we looked back at the old pictures and said, it's always been like that. But there is, and I don't know if we have a pointer, I don't think so, but I think I can move my, no, my arrow doesn't show up there. Sure, thank you. But there's a little bundle in the very bottom of that nerve, which has got a haze of white on it. In a moment, I'll use the arrow to illustrate it to you. The red, thank you. And right there, those fibers are just a little bit swollen, picked up very well by the instrument I mentioned, because they're carrying extra mitochondria. Unfortunately, this young man for many years came to us with just that. He was 12 at the time this picture was taken, and I think it was at age 14 that he lost his vision and went from carrier to affected in Brazil. So this is scientific. What is going on? In theory, the blockage of complex 1 that occurs in labors, and we often call it LHON, causes 2 things: less ATP, less energy, more reactive oxygen species. In this particular clinical event of labors, I think it's about the second, the reactive oxygen species, which is aggravated if you smoke and drink. That's a good place to get reactive oxygen species. Some of you have been my patients, and you know the story that I used to illustrate this point. And I was a visiting professor at UC Davis when they said, look, we want to show you a case. This was a 33-year-old woman who had suddenly lost vision. A little unusual given her age and gender, but it happens. And then she says, and here's my 9-year-old daughter. Well, that's really young. And then she says, and here's my 4-year-old daughter. And all of them had lost their vision at about the same time. And then further questioning revealed that they lived about a mile south of a giant tire fire where all the smoke had been billowing through their houses. And it took over 2 years to put that tire fire out. And so I thought, this is really classic example of the reactive oxygen species aggravating the disease. So I wrote to the EPA and said, do I have to go and take a jar, and if I do, and capture some of the smoke and send it to you, can you analyze it? And they say, oh, don't bother, we've done that already. We'll send you a computer printout of what's in that. And then in those days, the printers had the little pages of paper with holes on the side, and I got a ream about an inch thick with tens of thousands of chemicals in that. So I've given up the idea of which one is the bad guy. Smoke is bad. Now, one of the questions that I get, in fact, I got it yesterday while presenting to the main group of scientists, is why does it happen to the optic nerve? I just said that Leber's is sort of the mildest expression of mitochondrial disease, and yet one thing predictably goes, and that's the optic nerve. And the answer has to do with the fact that most of the body protects itself from some of these energy-related problems by using myelin, a coating around the fibers, the very thing that in fact gets inflamed in optic neuritis. But there is one place where fibers have to fire very rapidly and do a lot of work, have no myelin, and that's in the retina, because myelin's opaque and the retina has to be transparent in order for it to work. So that's why those cells are so particularly vulnerable. So I have a cartoon here, which actually was drawn by one of my medical students, although she's now a neuro-ophthalmologist. And I have a lot of fun because I travel around the world a lot, and I'll pick up a book in Chinese, but this cartoon always shows up in books in neuro-ophthalmology. And it shows the idea that complex 1 and complex 2 are donating electrons to complex 3, and if you block it at 1, it spills the electron, and that forms a reactive oxygen species. If you aggravate it with extra reactive oxygen species from tobacco and alcohol, and by the way, that doesn't have to happen. And in half the cases, there is no aggravation. It just happens on its own. But in this cartoon, it says the person had smoking and drinking. Add the reactive oxygen species to a critical threshold level. The mitochondria, they have an electrical charge, and that charge goes down. When the charge, which is normally about 200 or 220 negative millivolts, goes down to about 100, these little pores open up, and out come a cascade of chemicals that set off the death of the cell. And this happens in accordance with where the mitochondria accumulate. As we said, they tend to accumulate because there's no myelin in this particular section of the retina. So I'm going to briefly go through the next slides, which answers the second half of the question, which is why in the optic nerve is it these particularly small fibers in the very center. Clinically, we found this to be the case with the work of Pierre Barboni, who is one of the members of our team that goes to Brazil. It says it first affects the very small fibers that are called the temporal inferior fibers, and finally affects the supranasal ones. And I happen to have known from my anatomy that the first were small and the second were big. So we came up with a little math model, and we said, gee, the likelihood of a fiber anywhere in the body being involved with Leber's should be the number of nodes, that's the unmyelinated areas, times the length of the nodes, times the circumference of the fiber, because that represents the membrane that's working, divided by the number of mitochondria. And we crank through the math, and we discover the smaller the fiber, the worse the problem. And then we took our patients who had been generous enough to provide us post-mortem instructions to look at their optic nerves, and we oriented them so that on this particular picture, the left is where the smallest fibers are, and you see that in the mild case, it's just a little bit, but it gets progressively worse. In the more severe cases, everything's gone but the supranasal. We then did some analysis, and it turns out that the smallest fibers are the first to go, and the largest are the last. And one of my very good and very compulsive doctoral students made a map of the nerve where small is represented by white and large by black, and all the checkers have in between shades of gray. And I put it to you next to a map of a moderate case of Leber's, and the match, of course, is remarkably strong. So I think everyone now accepts that that math model does predict which fibers go when. So let's get to treatment. Having understood all these things about what causes Leber's, why is it that we haven't yet arrived at good treatments? And first we have to talk about the problems, and some of them have been alluded to already. If you've got structural damage to the optic nerve and everything's dead and you waited too long, and as Rustam says, sometimes it takes a year for the diagnosis, maybe it's too late. Should we be treating, therefore, people who have just converted? And there's a couple of issues there. And finally, what about treating all carriers? Well, we can't treat carriers on a lifetime of treatment unless we know what the risks are. And if we do develop drugs, we probably won't know what the risks are for many, many years. There have been many situations where drugs seem to be relatively safe and been put out there by the pharmaceutical community, only to discover that 5 or 10 years after using them, there are problems. It's one thing to ask somebody who is losing vision and has no good other alternative to take those risks. It's another to take a carrier who statistically is likely to stay fine forever anyway. And personally, I'm very reluctant to give them long-term risk in that situation. And by the way, the general statistic for the common mutation, 11778, is that half of the men and about 15% of the women will lose vision eventually. That statistic is both outdated and maybe irrelevant because to this group, anybody who's in this room knows so much about Leber's that they're going to modulate their risk factors and avail themselves of things that make the numbers much, much better than that. So that's all taking all comers, those numbers, and that includes people who smoke heavily. And if you're not smoking, you're not drinking, and you're doing other healthy things, then those numbers we call penetrance are much, much less than that. So before I talk about what works, which is in red here, I'll talk about what doesn't work in blue, and that is vitamins and coenzyme Q10 have failed all the clinical trials. I said that wrong. They failed some trials, but they weren't properly done clinical trials. So I think that there's still the jury out that even though there's no strong evidence they work, it's probably unfair to say they don't work. And many of you probably are saying, can't hurt. So taking reasonable amounts of these vitamins certainly is a reasonable thing to do. But we don't at this point have much faith to say that's going to solve the problem. Now, one of the things that theoretically should work, and one of the first things that was tried, is coenzyme Q10. That's the molecule that you may have recognized from my slide of not lies that takes the electron that should have gone from 1 to 3 and it carries it back to 3. So it should be a good thing. But why didn't it work? Well, the usual explanation that's given is it's a very hard molecule to move around. This long tail gets stuck in membranes, and so it doesn't get where you want. You take the pill, most of it comes out of you. That which goes into your blood, most of it doesn't get into cells. That which gets into your cells still has membranes to cross to get into a mitochondria. By then, very little is where you want it. So the Idebenone was developed in Japan as a molecule with a very short tail that therefore gets there, and we'll talk about that. And a third generation of the same class of molecules is Epi743 that has a longer tail than Idebenone but shorter tail than coenzyme Q10, gets in and still maybe works a little bit better. But we're also going to talk about gene therapy. There's 2 trials that we're conducting now, but also trials out of Miami. And finally, MTP-131, sometimes known as Bendavia. And I hate these names. It sounds like new cars. But when they put it in the drop for the eyes, they call it Acuvia. It's the same chemical. So Idebenone, does it work? And the answer is yes, a little bit over a long period of time in some patients. So it's a very watered down answer, yes. And these are the 2 papers that give the greatest credence to it working. One of these is a prospective trial, which is what we usually like. That's what Rustin was talking about, where you have some people get it, some people don't. Nobody, not only the patient, but the doctors don't know which group is which. So it's really done in a very good way. And our trial, which is retrospective, which starts off as a less compelling trial because it was, well, let's look back at the last 10 years of having given Idebenone and see what happens. Now, I should mention, by the way, that the most important authorships of both of these trials are sitting in the front row right now. And Valerio led our team with the retrospective trial, and Patrick was a heavy lifter for what they call the ROTOS prospective trial. So each trial went into this and got published in the same journal, probably for this reason, with some problems. But taken as a pair, they're complementary, because they show similar results. And we'll start with the ROTOS trial. 85 patients, 2/3 of which got treated, 1/3 did not. Nobody knew which group was which. They got a pretty high dose of the Idebenone. And as is usually required, the endpoint, the definition of success or failure is, did it improve visual acuity? And they did so at 6 months. And you don't want to look at this very busy slide, but I'm making just 2 points. The first is that the endpoint that they were going for in terms of improving visual acuity did not reach that p-value that Rustin was talking about. P equals 0.05. So in this most simplistic way, it failed. It didn't get there, and therefore, for example, the American FDA says, whoops, didn't make it, we're not going to make it available. But I'd like to point out 2 things. The first is that while that's a perfectly reasonable thing for us scientists to do, to say 5% chance of it happening randomly is as much as we'll tolerate, The number that is actually displayed there is 0.23, which means by random chance, there's a 23% chance that these events happen. That still means it's more than 75% likely it worked. So people don't think that way, and maybe rightly so in science, but one of my greatest criticisms is when people will look at this trial, and if they had just stuck with that number, say, this proves it doesn't work. That's not the case. It wasn't proven that it worked, but failure to prove is not proof of failure. The other thing is that when they went back and looked at the data, they saw other things that did reach that p-value of less than 0.05. So that, as I said many times, and I've been quoted, in fact, by the FDA, visual acuity is a shitty measure of this disease. And there are better measures, and the better measures showed a much better effect. So it strongly suggested that this was an effective, or at least somewhat effective, medication. However, because these weren't the primary outcome measures, the US FDA was unconvinced. And regarding these other things, essentially they're called post hoc analysis. It's the equivalent to retrospective. It loses all the advantage of this placebo-controlled trial, or at least some of the advantages. What about what Valerio and his group did? Our group. They looked at 103 patients, and these were patients that had been treated with different doses over periods of time, consciously aware they're on a medication, with the doctor knowing who got it and who didn't, all the disadvantages that Rustam said that we have when we do that sort of trial. But they were looked at over a period of 4 to 5 years, whereas the first trial was only 6 months. And look at this. This is what is called the Kaplan-Meier curve. And it basically says, over time, that's months on the bottom, what happens if you don't treat, what happens if you treat. And as you can see, the differences up to the first year are minuscule. No wonder they didn't see much of an effect in RODOS. But the further out you go, the more these lines diverge. And many of these patients showed significant differences at 2 and 3 years. So it takes a while for this effect when it happens at all. Well, I said there's another generation of quinone. This is the EPI-743 trial, a very complicated molecule that does funny, interesting things. And that's our paper that described only 5 patients with this. Since that, we've had a chance to do more patients. 12 patients have been done at Doheny in Los Angeles, 6 patients in the Brazilian group, and 3 patients in Italy. We've been following these patients out for about 5 years. And the results, I'm going to first try for the drama. This is one young man who showed no improvement after 1 year of being on the medication. Thank goodness the plan had been to keep it much longer, because 16 months later, suddenly started getting better. And as I show the visual acuities, it went from 2400 to 2030, which is pretty dramatic. And the visual field had a big scotoma in the middle. It became a small scotoma. That's a much better way, I think, of measuring this disease. That's one eye. What about the other eye? Left eye showed the same thing. 2400, 2020, big scotoma, small scotoma. This is 11778, the one that doesn't usually spontaneously get better, although it does once in a while. So was that what happened? I don't think so, because we found that there were, amongst the 23 eyes that we first analyzed, 7 that did the same thing. So 7 out of 23, or about a third, did show dramatic improvements. The upper graph is visual acuity, which I said was the shittier way of doing it, but everybody wants to see that. The lower graph is visual fields, which I think is a much more reliable way. It shows the same thing, even better. But there was another group of 10 eyes where things pretty much just stabilized. Didn't get better, didn't get worse. And unfortunately, a third group, 6 eyes, where notwithstanding the medication, it got a lot worse. I would have said 0 effect on those patients. So the 2 lessons of EPI-743 is that it takes a long time again, 2-3 years sometimes to fully play out, and nothing happened inside a year. And 2, it doesn't work on everybody. And in fact, in the majority, if it works, it only works some. So it's certainly not the answer. But I was very pleased. First of all, I have to agree with what both Rustam and Lissa said. This is the cusp of great things to come. And one of the things that happened that made me very happy was exactly this time of year, last year in Washington, D.C., where finally the FDA invited me to go and testify. And what I was testifying was not for a drug, but for an approach. The FDA wants to have visual acuity as the primary outcome measure. By the way, I argued against that and lost. It's still going to be the primary outcome measure. But they also wanted to have, as the standard way of doing it, what Rustam referred to as the double-blind placebo-controlled trial. One patient gets it, one patient does not, and nobody knows who's who. The computer does the coin flip. And even afterwards, the patient gets either the real pill or a sugar pill, and they don't know. But the trouble with doing that is a trial for Leber's, I argued, was both a very bad idea scientifically and a very bad idea ethically. From the scientific point of view, we were going to compare patient A to patient B, and there are differences. Some patients run a worse course than others. And so there's going to be a lot of noise in that, and you need to have very large numbers to get that to sort of iron out. And we don't have large numbers in Leber's. And the data that I brought to the FDA showed that although there's a variation in the first few months, because one eye goes first and then the second, by 6 months, there's extraordinary similarities in visual acuity, visual field, and the structural measures, the optic atrophy and the OCT measures between the two eyes. So wouldn't it be better to compare the right versus the left eye? The second thing was the ethical issue, I'm sure you'll all appreciate. Notwithstanding the fact the FDA did not approve Idebenone, people in this country avail themselves over the internet to get Idebenone. Is it fair to tell the patients, because if you're going to compare one group of patients to the other, you'd have to take them off the Idebenone, something that's doing them good? And finally, and this is a conceptual ethical issue, which I feel very strongly, but most doctors don't even get, which is each eye is not 50% of the whole. Your visual abilities are based on your better eye. So that if I had in-between vision, let's say moderate visual impairment in both eyes, and the devil came up to me and said, I'll make a deal. I'll blind you completely in one eye and give you back your vision in the other eye. I take the deal in a second, because I'm limited to my better eye. And if my better eye is 20/20, I can still read and drive and recognize faces fully. So if, in fact, we have a drug that might improve one eye, that's doing 99% of what we want to the patient. Flip side of that is if we have a drug that will years later turn around and bite you, it'll only do it in one eye. And losing vision in one eye may be sad, but it's not a catastrophe as long as the other eye is good. So it maximized the benefit, minimized the risk, and that argument prevailed. So the FDA says, okay, maybe we'll accept that sort of a trial going forward. So they're now, what we are doing, I'll start with what, of course, I'm enthusiastic about, is 3 new clinical trials for Leber's. One is gene therapy, injecting in the eye a virus that carries the gene that substitutes for the mutated one in the acute cases of 0 to 6 months. In one sense, that's maybe the greatest promise because there's been the least damage in the first 6 months. But on the other hand, in the very setting that I described, one eye against the other, it's injecting while there's asymmetry. You might get the better, you might get the worse eye because in the first few months they are different. The second gene therapy trial is 6 to 12 months. Maybe it's a little bit less of a window of opportunity, but it's also dealing with the symmetrical situation where the science will prevail and demonstrate itself very well. Thirdly is a group that you know as Stealth, and it's for more than 12 months when things are really stable and symmetrical, and it's using NTP-131. It's a molecule that sort of stabilizes the membranes of the mitochondria. Okay, you're absolutely right. Let me mention that right now. The 95% of the patients who have Leber's have one of 3 genes involved. The most common is 11778. When you get the gene therapy, you can't give a 11778 gene to a person who has 1444. So the gene that's been developed, both by the group that is working with us and by the group that's working in Miami, which we'll talk about both later, is just the 11778. So that's only about 60 or 70% of Leber's cases. So for those who have another gene, the gene therapy can't work. So anyway, all of these trials are working with this new FDA paradigm of saying you can randomize the eyes, not the patients, guaranteeing that every patient will have at least one eye getting treated. So the GenSight, this is the gene therapy that we're using. This is just their slide, and so I don't know why I had to have it, but it says that we're injecting the eye to try to treat the million ganglion cells that might still be there. They're not a million. And this is the point that Valeria wanted to make sure I said, which is here, which is that it only is for 11778, the most common mutation. But before I talk about our trial, a few words about John Guy. That's a picture of him there. He's at Miami. He works together with Byron Lamb. And before we go any further, he's the one who first had the idea of gene therapy. We've got to give him credit for that. He's the one who first thought of the fact that it could be done because it seems like it can't. And the reason it seemed like it can't be done is that gene therapy is putting a gene on a virus and infecting the cells with the virus. When that happens, the virus dumps its genes in the nucleus, and that's not where the problem is. And so at first glance, it's an insurmountable hurdle if you have a gene problem in the mitochondria. But he came up with the thought that you could get a targeting sequence, that you could deliver it into the mitochondria, which you needed to do, and work around that otherwise insurmountable problem. And when he did do that, which was about 10 years ago, most of us, I certainly will take responsibility, said, can't work. Sounds too good to be true. And here we are doing the work in different ways, so let's give him credit for having thought of that. So at Bascom Palmer Eye Institute, that's in Miami, John Guy and Byron Lamb are doing a study using the 11778. It's an open-label trial. It's not the ideal double-blind placebo. They did 5 patients, and they claimed these things. So this is a slide from their group. 1, the visual acuity is either improved or remained stable in all 5 patients. 2 patients got better, no serious events, and as I said, it's open label. So that means that the patient knows which eye gets the injection. That means that they know things that might give them a bit of a placebo effect. And in fact, I'm thinking that maybe one of the 2 effects could have been a placebo effect. But the other one, the 15 letters, that looks very, very real. It looks like it really got that one patient at least quite a bit better. Another group that's been using gene therapy is this group in China. They just came out with their publication of 9 patients, and at a period of 9 months. Some of them got better. The red lines are usually over the blue lines, but not always. And some of them didn't make sense because both eyes got better, like in this one here. And so there's some fishy and funny things, and some didn't have any effect. So it's out there. It's published. You've heard of it. I'm not sure what to say. I guess I'm going to suspend my belief or disbelief in this particular trial. It wasn't done the way that I think that we're trying to do the trials right. And then our gene therapy trial, which is sponsored by the French group, GenSight, they have a different way of trying to target the effects in the mitochondria. They first ship the gene into the nucleus, and from there it transports the product into the mitochondria. And what they have done is first get a proof in an animal that they could get the gene in, then proof in a petri dish that Leber's patients, Leber cells that get this gene do better. Then they went to monkeys. They injected it. Initially, they ran into troubles. The higher doses caused a lot of inflammation in the monkey eyes, but they were able to work out a lot of the kinks where they should have in non-human situations, and came up with the phase 1, phase 2, and thanks to Rustum, now you know what that means, way of demonstrating that it was safe in 2014 in France. And with the parameters they've worked out, we've agreed it's time to do a phase 3, which is the one that would show whether it works or not in the U.S. This is the phase 1, phase 2 trial, 9 patients, and sometimes the companies, in order to get others interested in them, will use that same data to say, look, not only did we show it's safe, but it worked. This is a slide they gave me permission to use. And basically, they've divided it into 4 lines. The bottom 2 lines are the patients who would not have qualified for the trial that I'm doing right now because it would have been either too bad or too late. And they showed that there's no difference between the 2 lines, meaning the injected eye and the non-injected eye. It didn't matter. Then the patients that would have qualified are these 5 patients here. And these patients had better effects in the eye, the green line, they got injected, and lesser effects in the one that didn't. And the differences are the same 17 letters, by the way, that the Miami group did. But these are just 5 patients. When we're talking about effects of random fluctuations, this happens in 5 patients. So this is why no serious scientist would look at this data and say, aha. But it's promising. But this is hopefully going to be when we can say, aha. Excuse me. Thank you. I've never gotten so many people saying bless you at once. So this is a trial that's being done in many centers. As you can see, we have 3 places in the U.S. I'm doing it, and Nancy Newman at Emory, and Mark Moster in Philadelphia. and Patrick right there is doing a trial in Valeria. So basically, half the people running those trials, again, in your front seats right here, and it's going to be really properly done. Randomized, double masked, sham control. That means that the patients will walk out. Well, let me tell you the way we do it. The patients who come are my patients first, so you become my patient first. We get to discuss things and establish that you'd be a good candidate, for example, having the right gene, and also that you understand the risks and benefits of the trial. Then, having established that, we do a huge day of examinations. Actually, it spills out and it's 2 days. Then you come back when the trial is going to be a few weeks later and have it done all over again to show that there's been no change from initial to later. Then you go across town. We are the Doheny Eye Institute, which is part of UCLA, but we're 30 miles away from the Jules Stein Eye Institute, which is also UCLA. So it's really nice. We have this firewall between us. We know what's going on, and they don't. That's one way of saying it. The other way of saying it is they do all the work and we don't, because they do the injection of the eye that is going to get the virus, and then they pretend to inject the other eye by pressing the needle up against the eye and creating a very similar little tiny hemorrhage on the outside of the eye, so it even looks the same to us. Then we see the patient again, not knowing which eye got injected, and then we follow it again. So with that situation, we're so separated from knowledge that there's no placebo effect that could possibly leak in. The trials run in two natures. One of them, again, fancy names that drug companies like, called the rescue trial, means less than 6 months, 36 patients. And then the reverse trial is 6 to 12 months, another 36 patients. And the recruitment is well underway. Here you see the institutes I just mentioned, and I like to say we are winning. We have the most patients recruited. And obviously, we all are trying to get as many patients as possible. We're well on our way. I think that at this point, there's almost 20 patients already enrolled in the trial. It won't be but another few months that we'll be able to get to the sort of numbers that are going to prove whether this works or not. So if it's less than 6 months, it's the rescue trial. 6 to 12 months, the reverse trials. This is the name of the virus, AAV2. This is the name of the particular subunit of that has to be put into complex 1 to make it work again, ND4, and that's the way it's being done, both, in all these centers I mentioned. What about patients who come to us and it's been more than 12 months? They qualify for the MTP-131 trial, which is going to run for 9 months and use this molecule that's over here. It's not clear how the molecule works, but probably has to do with the fact that there's a structural protein in the membrane of mitochondria called cardiolipin. Most doctors know about that because when you have autoimmune diseases, you have antibodies against this. It's very important at maintaining the alignment of the chemicals that move the electron along, and it gets bent when disease like Leber's happens. It gets misshapen, and that leads to these other elements not working well. The MTP-131 is a cage that locks around the cardiolipin and either keeps it from bending or gets it unbent. And here's the more fancy way of describing the electron transport chain and the way that the cardiolipin and the MTP-131 interacts with it and helps it. And the question is, can we shut down this overproduction of ROS that's part of the disease? And to the answer of that, I'm going outside of the field of ophthalmology, because Bob Kloner has done some wonderful work, very simple structural work. He takes rats, he takes the suture, and he ties off the left coronary artery, the main one for a rat, and he gives them a heart attack. And then he measures the size of the heart area that is infarcted. And then he does it with and without the Bendavia, which is the name they give to the MTP-131. And these are the graphs that show that if you pretend to ligate the artery, you end up with 1, which means 100% of the heart okay. If you ligate it, 50% of the heart is destroyed. If you put in the MTP-131, you get about 80% of the heart preserved. And not quite normal, but it's obviously done a terrific job at reducing the size of the heart attack. And this work is just, extremely compelling. And now, yesterday, we got to hear about another piece of work done by the same group with the same chemical, and it's called the Power study. Again, it's not ophthalmology. It's about patients who have significant myopathy, a weakness of the muscle due to mitochondrial dysfunction, and it's got a way of measuring how bad it is by seeing how far can they walk in a certain period of time. And they took the group of people with them and they split them off again with the placebo. Some didn't get the agent. It was given by IV transfusion, so they can't tell if it's saline being transfused or the agent. And they gave it incrementally greater and greater quantities. In my mind, this quantity here of 0.01 is probably pretty close to placebo too. But then they got a moderate dose and then a high dose, and they got this escalating series of doses, and they found this. And I find this extremely convincing, because when they gave the placebo, they didn't get much of a benefit. When they gave the low dose, they didn't get much of a benefit. When they gave the medium dose, they got a lot of benefit. And when they gave the high dose, they got even more benefit. So this is called, dosing. I'm sorry, I can't remember right now the buzzword for it, but it's a dose effect. The greater the dose, the greater the benefit. That's very real. The placebo part is also very good too. So it's a very convincing thing that they could walk further in meters depending upon how much they were getting. Then this is a further analysis from a slide they provided me, and I have permission for these slides because apparently they're quite confidential. But what they also found out is that the slope of the curve, how much better they got, depended not only on what dose they got, but on how bad they were. And it's a very convincing thing to me, because when the patients had the least disease, they got the least effect. When they had the most severe disease, they got the most improvement. So this is, again, the way that nature and biology should be, and especially in a placebo setting, very impressive. And finally, there are a couple of areas that Lisa said I could touch on, and I've decided to call them future directions because we're not ready to talk about the clinical results yet. One is the idea of using estrogens to create more mitochondria. One of the reasons that women seem to be doing much better than men with this disease is that they have natural occurring estrogens. And you can get estradiols both from your diet and from different ways. We're not ready to talk about the effects of that yet. The other is that we are now understanding better the carriers, and we can be classifying them and perhaps see who's at most risk. I am not talking about mitochondrial replacement therapy because it's not quite happening, and also because both on an ethical and legal basis and in terms of the science and medicine, most of the work that's been done in that has been done at Newcastle, England, and there's a guy from Newcastle, England, so I thought if anybody should ask questions about it, they should be directed to Patrick. One thing I did want to finish with in mentioning, which is stem cell transplants at this point are scams. Maybe one day there'll be a wonderful new therapy for this, but they're not now. And since there are places, particularly in China, that advertise it, and what they're really asking for is your money, and you spend your $50,000 and go out there and have your own stem cells transplanted into your spinal cord, which is the wrong place anyway because nobody has eyes in their spinal cord, it really isn't going to be a benefit, and I would ask you not to do that. If you have $50,000 to spare, I certainly know all these researchers are better places to put them. So I'm going to finish by saying I think that I've seen over my lifetime about 600 patients with Leber's, and I've watched, obviously, the tragedy of young adults who go blind. I've been unsatisfied with the success that we've had with the 2 quinones, although it does work a little bit, but it only works on some, and it takes a year to work. So this is the year that I'm going to say I'm excited. I think with all these new therapies and clinical trials about to come online, we're at the cusp of great things. And thank you very much for having been our partner in that.
Thank you so much, Dr. Sadoun. Thank you. So next, I'd like to take a few moments for questions and answers. And I'd like to ask a couple folks to join these folks up on the stage here. One of the great things of having this meeting with the UMDF is that it does attract people from around the world talking about mitochondrial disease, and since they're here, we get to take advantage of them being here. So in addition to the folks you've already heard from so far, we have Dr. Patrick Uiman, who's a neuro-ophthalmologist based within the Wellcome Trust Center for Mitochondrial Research and the Institute of Genetic Medicine at Newcastle University in the UK. He also holds appointment at the Newcastle Eye Centre at Moorfields Eye Hospital in London, where he runs a specialist clinical service for patients with mitochondrial eye disorders. And from Italy, We have Dr. Valerio Corelli, who's a researcher in charge of the Laboratory of Neurogenetics, Department of Biomedical Sciences and Neuromotor Pathologies at the University of Bologna, Faculty of Medicine and Surgery in Bologna, Italy, who's published over 200 papers. And with him is Chiara Lamorgia, who's a neurologist in that, also in Bologna, Italy. So if people have questions, please raise your hand. And sorry, but please wait for the microphone because otherwise on the video we won't have the question captured.
You mentioned that.
So I'm going to hold a cardboard. It's not that I don't want to see you, but otherwise I can't see you.
Yeah, you mentioned that myelin is the one agent that's protecting the eyes, and when the protection of myelin is not there, that's when we see this mitochondrial effect. Are there other body parts that also lack the protection of myelin? For example, in my daughter, I see renal tubes or nerves having similar damage to the eye. So is the myelin protection lacking in other body parts also?
Sure. That's a great question. Let me rephrase slightly. When we did our math model, taking into account the myelin, the prediction was that the very first part to go would be the smallest fibers of the optic nerve, and then in a wave, just like we see the rest of the optic nerve. The next thing that it predicted is that the longest fibers of the peripheral nervous system should be involved. And so these are the ones that go all the way down to your feet. And most patients with Leber's don't get those involved, but a few do actually. But we had the opportunity of biopsying from some individuals that area and looking at it, and it is involved. So the answer is to get clinically significant is rare, but to get some involvement of the peripheral nerves, just as you would suggested, it does happen.
One second. Since what you're doing now just involves the 11,778 people, do you think what is being done with them will be applicable to the other kinds of mutations? And if not, are there going to be studies done with people with the other mutations?
I apologize for taking the microphone, but I got the answer to this one. First of all, the genetic trials are limited to 11778. But for example, there are other trials going on that do apply to all the mutations. Second of all, both the groups that we're talking about, the trials I'm doing, the GenSight has already started working in animals on the 1444, and then they'll follow that with 3460. So for the big three mutations, I think that within a year they will have the gene and the capacity to do it there too. As for the MTP 131, if it works in one, and we're choosing one just to be purist scientifically, it'll work on the others, and then it'll be immediately applicable.
I was wondering, in the clinical trials that are being done, does it matter whether the person has lost his sight 5 years ago, 10 years ago, or has now been living with it for over 20 years? Does it matter?
Maybe, but we've been fooled by that. Initially, I thought that the window of opportunity was measured in hours, and then as I learned more in this field, I said, well, this seems to be days, and I've shown you data that says that it's at least 2 or 3 years. So I don't know what the backside of that is. I know that Valerio has had experience with some patients being put on Idebenone. How many years out?
Well, we had a few patients being treated in the very chronic stage, and occasionally you can see little improvements, but most of the cases you don't see much happening. So the point is, depends really on how many fibers and cells are still remaining into the system that you can work with, even if the disease is 10, 20 years old. So I would not, I mean, it could be tried something, but I would not be too much optimistic that we will see some results. I don't know if I'm clear.
Yeah, I think that the answer is longer is more difficult, but there's no absolute back end to this. But let me also say that there's work being done for those issues as well. And Professor Sahel, who was in Paris, and by the way now is in Pittsburgh, come to the US, has some preclinical studies which I think in about a year or two, not that far from now, would be applicable to people who have complete optic atrophy. So we'll see. The science is marching on all fronts.
Yeah, if I can add something, the options that Alfredo presented are those which are the closest and the ones that are actually in action right now, the closest to be in action. But theoretically, there are other options that are still there. So mitochondrial biogenesis and the possibility to use phytoestrogens to boost the increase of number of mitochondria, which seems to be a good thing in terms of compensating the Leber's mutation, is one avenue that still has to have a company promoting a trial, but there is good preclinical data in that direction. And I just heard yesterday, I'm aware that for the little subset of patients with heteroplasmic mutations, there are now tools actually to shift the heteroplasmy within tissues, and that's going to be something in the next future that could be another option. I mean, it's not going to be applicable to many patients, but the little subset of people that has heteroplasmy, so a mixture of mutant and normal DNA, there are other strategies that could be actually placed in action.
I thought I heard from Dr. Sadoun that although there is a misconception that the symptoms stabilize over the first maybe a year or two, perhaps over the first year, in actual fact, I thought I heard you say that in fact they don't, that the symptoms can continue to become aggravated over long periods of time, and in fact it may never stabilize. Did I hear you correctly? And if so, can you address that?
You did, but we're speaking in this setting of the Brazilian patients. So under the circumstances of not only not treating, but of probably having suboptimal nutrition and smoking and drinking, it can continue. Yes, ma'am.
You talked about estrogen. So is that something that both affected and non-affected should be supplements and that type of thing?
We need to be careful with that.
Okay, yeah.
Estrogens, as they are, cannot be administered to males, for example. I mean, there are a number of risks besides some interesting side effects. So, well, the idea is to develop some topical administration drops, and with estrogens that are preferentially being directed to one of the receptors, so avoiding some of the side effects we were talking about. And theoretically, estrogens should be dedicated to preventing the disease, because boosting the mitochondrial biogenesis seems to be the right thing in order to maintain unaffected the eyes. But as Alfredo pointed out, we need to very carefully plan a long-term trial, well-designed, because giving a drug to a carrier, to an unaffected carrier, and then if he gets the disease, we will get in trouble, because you never know if you actually did something wrong. And so only a well-designed clinical trial with awareness from the part of the patients that is getting enrolled of the risks and the acceptance of the entire scenario is the right thing to do. We cannot experiment here and there just like this. Yeah.
Dr. Diana, could you talk a moment about mitochondrial replacement techniques?
Sure. And as Alfredo mentioned in his great talk, what is really pleasing about the science that all these various strands are maturing, and each one of them are providing options that might be more applicable to certain groups of patients. Certainly the work that has been pioneered by Doug Turnbull and his team in Newcastle is now again reaching scientific maturity, where about 2 weeks ago they published a paper in Nature showing that the technique was safe in terms of the embryo growing to a certain stage. And as you will be aware, last year the law was changed in the United Kingdom that would allow this technique to be applied to prevent the transmission of various mitochondrial DNA mutations from mother to child. So essentially, at this point in time, things are at a reasonably advanced stage of development, both in terms of the legal framework and also in terms of the scientific preclinical work. But of course, there's more work that is being done at the moment to make sure that the technique is safe, because it will be, a first in man. This has never been done before. And again, there will be a series of discussions with various regulatory bodies and also with patient groups and also other stakeholders as to which group of patients the technique, this mitochondrial replacement therapy technique, should be made applicable for. So there's been a huge amount of progress made over the past 2 or 3 years, and you also heard a talk yesterday from the other group in Oregon who's doing a slightly different technique but the same principle. We just need to follow the story and see when it will reach a stage when people are happy that the science has reached a stage where we can actually apply that to a female carrier of a pathogenic mitochondrial DNA mutation.
Great, thank you. And I think at this point I'm going to wrap up the Q&A.